So good afternoon, everyone. My name is Frank LaFurla, and I'm the Dean of the School of Biological Sciences, and it's so nice to see such a nice turnout for a very fascinating talk today. And it is my distinct pleasure to introduce one of my colleagues, Dr. Craig Stark, who is a professor of neurobiology and behavior, who is one of the world's leading experts on brain imaging. Truly a pioneer in this area, has developed some very sophisticated tools and techniques involved in brain imaging, and has been able to use this to study a range of biological processes from Alzheimer's disease to autism to depression, even understanding things like video games and what happens to your brain during that. And now he tells me he's also going to understand what effect radiation in space has on brain activity. So it's very cool. And his topic obviously focuses on the brain, which fits into our foundational theme of the school, which you could see on these posters, relates to mind, body, and world. And the basis for that is pretty simple, because we've come to realize that life is interconnected. And certainly for most of us, to have a healthy mind, we need to have a healthy body. And to have a healthy body, we need to have a healthy world. And so that's why our theme is just mind, body, world. So with that, let's welcome Professor Craig Stark.
Well, thank you, Dean LaFurla, for that introduction, and thank you all for coming on out today to hear a little bit about the kinds of things that we're doing at UCI with neuroimaging, and the kinds of things that students are helping us out with when it comes to research. Because the research I'm going to be presenting today can't really happen without participation of students here on campus. Sometimes those students are then the ones who hop in the scanner and actually have their brains scanned, but most of the time it's that there are students, whether they're graduate students or undergraduate students in my lab and other labs here on campus, really helping out to actually get the research to work. So first I want to just give a quick overview as to what UCI is doing in terms of neuroimaging and the kinds of different facilities we have that different labs are actually using. And I want to bring this up really because just a couple years ago, with the leadership of folks like Dean LaFurla, but then also Dean Stamos in the School of Medicine, Dean Maurer in the School of Social Sciences, we put together a collection of neuroimaging resources for people to actually be able to use. So for example, we have this grand vision called the Campus Center for Neuroimaging, and it brings together our previous neuroimaging center that has a PET scanner and an MRI scanner, and the brand new one that I'm going to be talking a little bit about today called FIBRE, or the Facility for Imaging and Brain Research. And we're currently building a state-of-the-art small animal scanner so we can do things like take experimental mice and rats and be able to do the same kinds of scans on them. So just about a year and a half or so ago, this was a big event here on campus. We had a gigantic crane over on one of our buildings, and this right here is the MRI scanner. It's several tons worth of $3 million worth of equipment that got carefully lifted all the way up to the height of the building, dropped down into a little hole, gradually lowered into a hole, not dropped into the hole, and installed as really the cornerstone of our brand new imaging facility here. This scanner can take all sorts of kinds of pictures that we use to try to actually understand how the brain works. Has anybody been in an MR before? Banged up a knee or something like that? Yeah, okay. So you know you lie inside this tube and it makes all sorts of big banging noises, and out of that we can get some pretty cool pictures. We can get pictures that show what the brain actually looks like, or the knee or the shoulder. We can do things like say, how is the brain connected? What's the circuit diagram, the wiring of somebody's brain? We can see where is their activity in the brain, where are the neurons actually firing, and we can also start to get at some of the biochemistry as we try to look at different brain metabolites and see where they are actually deposited in the brain. But I at least wanted to give you a little bit of an idea as to how this machine works, because it's a really pretty incredible machine. If you think about it, you're lying inside of a tube, and there's a gigantic magnetic field that you do not feel at all. And with this huge magnetic field, and it's all just that big magnet, we're going to take a picture of something inside of your body using magnets. And it works by you lie inside of here, and I mentioned it's this huge magnetic field, and what it does is it gets all of these protons, these hydrogen atoms like in the water inside your body. It turns out that they're a little bit like a compass. They have a little bit of a magnetic field, and so you put a big magnet there and they're going to line up. We line them up on that magnetic field. We get all of them sort of lined up that way. We then apply another little magnetic field. We bop things off to the side. We get these guys spinning at a certain rate. We do all sorts of fun math. And at the end of it, we get an image that looks like this, which isn't all that exciting, but you do a little bit more math on that image, and it turns into something like this. It's pretty darn cool. And as I mentioned, it's all happening just with a gigantic magnet. And with that, we can then tune the scanner up to look for all sorts of different things. So if you clinically had an MRI scan, they might be looking for something like this. Here, a person has actually had they have something wrong clinically. This is being used in a diagnostic purpose. As you can see, there's a big sort of shadow over on this side of the brain here. Something is clearly going on. It turns out this is a tumor, a diffuse tumor. We can tune the magnet up, though, in ways where it looks like this, or we can tune the magnet up to be able to show something very bright and say, okay, no, this is where it is. And a lot of MRI is done for that, for diagnostic purposes. But we get to use it for all sorts of fun research purposes. From looking at the basic science as to how the brain is wired up and how it's structured to things like, how are the neurons firing inside the brain? Because it turns out that as neurons fire inside your brain, so right now you're listening to me, hopefully you're listening to me, there are neurons in your auditory cortex that are firing, that are helping you decode what it is that I'm saying. And when those neurons fire, if I stuck a little electrode inside your brain, and I know you don't want me to do that, but if I did, we would see that you could get these neurons to fire a whole bunch as you're listening to me. And what happens as a result of that is as they're firing, they're using up energy, and they then need more nutrients. They need more oxygen. And so the body responds by bringing all sorts of fresh oxygen into that little spot in the brain, and we get a little signal that comes out that looks like this. Because it turns out that blood with oxygen has a little bit different magnetic property than blood without oxygen. And so we can then do things like stick little colored blobs on the brain to say, hey, this is a spot in which a little bit more activity was going on. And this is called functional MRI. And so we can tune our scanners up to see these kinds of things. Where is the brain more active when you're doing one thing versus another? And this has become a very staple tool whenever you hear on the news, researchers have found that patterns of brain activity are different. This is usually what they're actually doing. But that's only one of the things that we can do. We can look at the activity here inside the brain, but we can also look at the size and shape of structures in the brain. What happens as we age? I'm going to tell you a little story about aging in just a little bit here in terms of how the brain is actually changing. We can also, as I mentioned, look at how different regions are connected to each other, really the wiring diagram of the brain, and see, does that change with disease? Does that change with age? Does that change as we develop? And the answers are yes, yes, and yes. And we can also, this is a neurobiology program that I'm in, we can start to get at things like individual neurotransmitters and see how they may be changing, again, with damage, with disease, with the development. So I mentioned that we have this brand new facility, and I got people like Dean LaFurla to fork over many millions of dollars for this facility, and thank you again. What makes this scanner so special? It's actually the best scanner, the best MR scanner that money can buy right now. And I suckered him into actually paying for it, so that was a great thing for us. Well, one of the things that makes it so powerful and so great and so exciting for the research community here is how quickly we can go. So when we take MRI images, you lie inside the tube, and really what we do is we take an image here in the brain, and then here, and then here, and here, and here, and here, and here, and here, and we build up a full three-dimensional picture by taking individual slices. Now, our scanner we used to have could do that pretty well. We could do that in about 2 seconds or something like this. Because you would go and you would do one slice at the bottom, and you'd go all the way to another slice at the top. This guy here, I can actually grab 16 slices simultaneously. I've done the entire brain in about 1/4 of a second. All right. That means I can get that much more data. I can get that much more resolution as to how activity is bouncing around and flowing inside the brain. So speed is one huge thing. Another huge thing is the clarity of the image that we can get. So I kind of liken this to saying, yeah, we used to have a scanner, you have another. All right, if you had the great shot, you finally got all of your family together, everybody's smiling, everybody's happy, maybe you have the Grand Canyon or something in the background, there's an eclipse going on, this is the ultimate shot, are you going to grab your current generation of phone or say, you know, let me grab that 10-year-old one over here? Well, no, you're going to grab the new one because you know the picture is going to be better and sharper and cooler. So here is a previous generation kind of scan, and here's one that we can get out of the new facility. And if you zoom in here, this is the structure I'm very, very interested in. It's called the hippocampus, and here you can see a lot more than you can there. And this is the kind of thing, again, it's sharper pictures that we can get. We can also do new cutting-edge novel methods. So this is a study, this one actually was done on this same model of scanner, but it's looking at things like if you have somebody who has a traumatic brain injury, this is actually from a sports concussion study, being able to see that we can look, instead of just one tiny little spot in the brain, we can look throughout the brain and be able to see the chemical composition inside here. We can also go through and do fancy new kinds of ways of mapping out the connectivity, the underlying microstructure in the brain. We can pull this off because of the new facility. So let me just give you an idea. So now that we have this, what are people using this here for at UCI? So I'd asked a number of our users a little while ago, tell me a little bit about some of the things that you're actually doing in here. And here are some of the things that people actually related to me. So for example, how does the brain change with age and how does it relate to cognitive changes? That's one going on in my lab and in a number of other labs. What can we learn about diseases like Alzheimer's disease and their progression? By studying the brain between people who have Alzheimer's, early stages are likely to get Alzheimer's, or even as we follow people longitudinally in the Alzheimer's Disease Research Center here and be able to see how this changes. How does the fetal environment actually affect development of the child? I'm going to show you a slide on this one in just a minute. It's absolutely incredible. So things like also how can we observe the neural effects of interventions such as those that might reduce adolescent risk-taking behavior or addiction? How does the brain process language? How do different brain systems work together to give us memory? Do interventions like brain training affect how the brain processes information? So let me give you an example of just a couple of these. This is the work of Claudia Buss and Gerard Rasmussen. This is a study looking at child development that starts off in utero. So they scan mothers who are pregnant and they're looking at the environment that the fetus is developing in. So over here we have a scan actually, and it's looking at the liver and looking at actually the amount of fat in the liver. So when they're looking at the developing environment that the baby is growing in, then they track these children after birth and then can see things like, hey, you know what? It turns out that if you affect this, the thickness of your brain's cortical sheet is actually changed. So trying to understand development from in utero through childhood by following people using this. One of the huge things that they need to be able to do is scan very, very quickly because everything is moving in a, in this in utero environment. Another fun one here, this is the work of Alyssa Brewer. And in her study here, what she's doing is she's looking at how the brain processes visual information. Because most of us may think that maybe babies, as you're growing, the brain figures out how to wire itself up and figures out how to see the world, and that absolutely happens. But it turns out, right now, as you're listening to me, your brain is still tuning itself. It changes even in adulthood. You are constantly adapting to the world that you're in. And she did a really clever experiment here. She took people, stuck them inside the MRI scanner, and mapped out exactly the structure of how different parts of visual cortex help the person see the world. Person by person, she comes up with these very, very detailed maps, and then she had people wear these goofy glasses. What these glasses do is they actually flip your world. Literally, they flip your world. So left becomes right and right becomes left. And yes, when you put these on, initially you are banging into things and you have to have somebody around you to make sure you don't trip and fall and hurt yourself. But it turns out that after a week or so worth of doing this, you've adapted. You've learned how to see the world differently by wearing these glasses. So she stuck people in after 14 days then of doing this. And what we're seeing here is this is normally then saying, okay, look, one hemisphere is responding to the stimulus and the other one isn't. This is what you were like beforehand. And after you've been wearing these glasses, now we get it so that both hemispheres are responding to this. So you can actually see how the brain adapts and rewires itself over time. So those are a couple of the things that are going on in some other labs. I wanted to give a little bit deeper dive on, I'm just going to use my stuff because I know it well, as to how this kind of technology can be used to help us understand something that we're all facing here, aging. We know that aging leads to problems in memory. Why? Can we start to understand what's going on in the brain that leads to this? Can we then try to determine what's different between typical aging and something like early stages of Alzheimer's disease? And can we figure out something we might be able to do about that? So I'm going to try to tell you a little bit of story on that really relies heavily on neuroimaging. And I'm going to be telling you a story about a brain structure I've been studying now for decades called the hippocampus. So here's an MRI scan. And if we rotate, one little animation there messed up. If we rotate the brain around here, go all the way into the middle, and then come back out, this structure right here is called the hippocampus. If you've ever seen any kind of documentary where they talk about memory in the brain, you've heard about the hippocampus. It's our favorite structure. Well, my favorite structure. Yes, I have a favorite structure. I'm a geek. And yes, that was my head, if you hadn't noticed. If we took it out of here, it's called hippocampus because when you look at that, you say, oh my gosh, it's a seahorse. Of course, it doesn't normally have the seahorse on there. It's hopefully helping give you that little vision. But we've learned over many, many years that what the hippocampus does is it's critically involved in certain kinds of memory. If you want to be able to remember what happened to you yesterday or even this morning, it's your hippocampus that's really going to be able to help you out. In particular, if you want to be able to remember any details of that sort of thing, you're going to be relying upon your hippocampus. We've learned a bunch about how this actually does it. But one of the big questions that we've had is, how does this actually interact with what's going on in aging? Because in aging, we know that there are all sorts of problems with memory, and they line up a lot with the kinds of things that we think are going on inside the hippocampus. When I test older adults, what they have issues with in their memory is memory for specific events. They may remember, oh yeah, I remember hearing this story about this. Who told it to you? I didn't get that detail, or at least I don't remember it now. It's this level of extra detail that we see seems to decay and have problems as we age, even outside of Alzheimer's disease, so in typical aging. A bunch of things, by the way, I don't want to just paint the picture that everything gets worse with age. A lot of things are perfectly fine as we age. So vocabulary is perfectly fine. What we call a simple gist or familiarity kind of memory, the memory that something happened, turns out that's not changing with age, but remembering the details of that event, that is what's changing. But what we wanted to try to see is, are these kinds of things that are having issues with aging, are they a result of changes in the hippocampus? And so my lab set out to try to get at that, and one of the things that we did, just want to give you an idea as to the kinds of ways we sort of probe this. One of the things that we did is to come up with a task that lets us really index this very well. This task is now actually used in a number of very large clinical trials, including this one, the A4 trial. It's about a $350 million clinical trial looking at Alzheimer's disease and how we might actually slow its progression. The task is very simple. I show you a series of pictures of color objects, like you might see this four-leaf clover, you might see this seahorse, and later on I can test you with those exact things. I can give you this four-leaf clover, and I can give it to you again. I could give you something you haven't seen before. And this kind of memory is a pretty simple memory. Have you seen any kind of clover like this? Have you seen this rubber duck? Yeah, I've seen the clover. No, I haven't seen the rubber duck. And it turns out that does not change with age at all. It does change as you're leading into the early stages of Alzheimer's disease. However, when you take a look at memory for the details, like maybe I showed you this seahorse and I test you with this one, well, okay, for the students here in the audience, you're going to be able to pick up on that pretty well and say, no, it's a different one. For your parents and grandparents, they're going to be like, yeah, it's the same seahorse you showed me before. All right, that's the problem that we see happening with aging. So is this, though, a result of something going on inside of the hippocampus? Well, to get at that in people, we're going to have to be able to peek inside and look at their hippocampus, and that's where neuroimaging comes in. So I mentioned before, looking at the circuit diagram, the wiring of the brain. We do that by putting you in the magnet and actually trying to jiggle water molecules around inside your brain. It's kind of crazy. You're lying inside the magnet, and I try to see how well I can move water molecules. Why? I do that because, well, let's say that you're a water molecule, and you're living inside of, there's this stuff called cerebrospinal fluid. It's basically glorified water inside your brain. You're in an area in which you've got water everywhere, and you don't really care about what direction you move in. But if you were in a thin little straw, you may be perfectly happy moving along the straw, but you wouldn't want to go across the straw because you've got the outside of the straw that'll be preventing you from moving there. Why the heck am I talking about straws and things? Okay. Well, the wiring diagram of the brain, you have neurons over here that are trying to talk to neurons over here. They send fibers that are axons of their neurons. They send a whole bunch of these fibers that go off from one part of the brain to another. And those fibers are like this. It's a fatty substance called myelin that's around them, and in between there are little tiny gaps where you're going to have a little bit of water. And that then will move along the fiber and not across the fiber. So if I can go and try to jiggle water in a whole bunch of different directions, I can see which direction is this fiber pointing in. Is it a nice tight fiber bundle, or has it gotten kind of all loose and weird and mushy? And so I can use how well the water moves along this fiber to decode the strength of that, the integrity of that kind of connection. So it's called diffusion tensor or diffusion weighted imaging. Here's a little hippocampus, here's the entorhinal cortex, and these little hot spots are the actual connections between the hippocampus and the entorhinal cortex. This is some of the connectivity of the hippocampus. And when we do something like this in our lab here, we can come up with this nice little signal that is the integrity of this connection, this input to the hippocampus, and we can see things like, hey, you know what? It goes down with age. It gets worse. As we are aging here, the strength of this connection goes down. A lot of things go down and get worse and saggy and stuff with age. All right, well, turns out also, though, that how strong this connection is correlates very, very nicely with how well you can do my memory task and not other kinds of tasks. So how well you can remember the details is really tied to the strength of the connections you have in there. And it turns out, by the way, that some of the people who are doing really, really well on the integrity measure are older people. And so if you happen to be older but have a really good set of connections in your hippocampus, you're still doing really well. If you happen to be younger and have a really pretty poor connectivity in your hippocampus, you're doing poorly on the task. So aging leads to some issues with it, but we all are different. There are individual differences amongst us. And if you've got a good hippocampal circuit, you're going to be doing well on this kind of memory task. So we're using this to try to help decipher why is it that some people age very gracefully and other people not so much. Another kind of thing that we can do inside here is really look at the underlying biochemistry of the brain. So you've probably heard of neurotransmitters, glutamate, dopamine, GABA. These are ways in which neurons communicate with each other. They send these little chemical signals to each other. And we can actually go and look inside the brain here and try to decipher underlying inside some spot in the brain how much of different kinds of brain metabolites or neurotransmitters do we actually see inside that spot in the brain. So if you've seen on like a CSI type show, they'll like take some little like paint chip or something like this, they stick it in a machine, they get some weird peaky squiggly thing like this out and they say, why look, this has to be from a 1987 Chevrolet because look at the paint signature. All right, it's actually pretty much that same thing, except we're doing it with magnets inside your brain. And instead of different kinds of paint, it's different chemicals inside your brain. So for example, here's this one NAA, here's glutamate, here's another part of, here's creatine, here's choline, here's myoinositol. They each have their own little chemical signature. And we can get this little spectrum, this black line, out of the brain, and we can go back and solve and say how much of each of these different kinds of chemicals were inside there. Because they all have their own little chemical structure, they resonate a little bit differently inside this magnetic resonance imaging scanner. And when we do something like that, we can see that, you know what, in the hippocampus, this thing called NAA declines pretty solidly here with age. Other regions not showing this kind of decline, but we can actually track that this is going on with age. We can see things like glutamate, neurotransmitter here. Glutamate, in aging here, actually is then predicting how well you can do my memory task. If you have high levels of glutamate, you're doing very well on memory tasks. We can see this kind of thing. This is seeing it the first time a couple of years ago in a publication from our lab. and just as of about 2 weeks or so ago, we nicely replicated it. So we can see that aging is leading to a decline in the hippocampus, but again, there are differences in how we all age. And those of us who still have very high levels of glutamate in here are actually doing well on the memory task. And no, by the way, I don't have a pill you can take to increase glutamate inside your hippocampus. Sorry. But the first take home from all of this here is that, yeah, our memory system does decline with age. It's not overall, it's specific kinds of memory. And it seems to be the kinds of memory that the hippocampus is involved in, that's really what's declining with here. But to truly understand this, we can't just say, yeah, it seems to be that kind of memory, so that must be it. We're scientists. We have to go through and try to see, can we actually tell a good story? Can we see that there are alterations in the connectivity? Can we see that the activity changes? Can we see that maybe the brain metabolites change? And can we tie these all together and say, yeah, this is why your memory is getting worse? Because that's the first step in trying to come up with any kind of treatment, any sort of cure. We have to understand how the system normally works if we have any hope of actually trying to fix it. I mean, right now, there's probably a 737 over at John Wayne Airport that's broken. And my guess is none of us here can go and actually fix it. Now, there are people who can because they know how that engine, how that jet plane actually works. The rest of us, we have absolutely no idea. One of the gigantic problems that we as neuroscientists face is there is no manual on the brain. We're having to reverse engineer and figure the system out as we go along. All right, so the first thing, as I say, is that, yeah, we can see these kinds of changes that's going on inside the brain, and we can start to then understand how things like the hippocampus work and how it changes with age, and this can lead us to some ways in which we might do something about it. So I wanted to tell in the last little bit here a story about what we may be able to do, and a lot of the students in the audience, you're really going to like the answer that I come up with here, so don't fall asleep on me yet. All right, so one of the things that decades worth of neuroscience has shown us is if you take a mouse or a rat and you let this rat live in what we call an enriched environment like this, where it's got these little tubes to run around, it's got some toys, it's got some areas that it can explore inside of its world, it's got a little running wheel maybe to exercise on. These mice and rats here do a lot better on memory tasks than rats or mice that live in a box with a couple of their buddies. It's a safe box, they're not stressed, but there's not much to do. There's not much to sort of push themselves to learn and figure out and do. So we call this environmental enrichment, and it does a whole number of things for your brain. One of the things that it does for your brain is it increases this thing called BDNF inside the hippocampus, brain-derived neurotrophic factor. There are other trophic factors that it does as well. And also it actually lets new neurons get born into your hippocampus and be incorporated into the memory circuit. That's pretty cool. Only 2 spots in your brain where, as an adult, you're getting new neurons, and one of them is inside the hippocampus here. The other, by the way, is the olfactory bulb, which helps you smell, because the ends of those neurons are exposed to the world and they're continually dying off as a result, so we need to keep making new ones. It's called neurogenesis. So in any case, we see that in mice, you get extra neurons born into the hippocampus, and it turns out, by the way, that the more mice go and explore a world like this, the more they get new neurons inside their hippocampus. So there's something about, even though these are all genetically identical mice, those who went and engaged and explored their world more, we would call it lead a more cognitively engaged, active lifestyle, actually did better in terms of the number of neurons. And as we also increase the number of neurons, they do better in memory tests. So, question that my lab had is, does any of this apply to us? I mean, it's fine, it's great to use animal models, but at the end of the day, having really, really smart mice, or mice that don't get Alzheimer's disease, I mean, that is great. He's a pioneer of making animal models of Alzheimer's disease to understand Alzheimer's disease, is Dr. Laferla, and that's a great thing to do, but at the end of the day, we want to be able to translate it to us. So the mouse can't be the end goal. So how can I try to see if something like this enrichment actually works in us and helps our memory and helps our brain? Now, this is not my lab. My department chair was actually a little fond of the idea of trying to have something like this for my lab. But after I asked for the scanner, I figured I had to at least wait a few years before I asked Dean Laferla to give me this for my lab. My lab looks a lot more like this. This is actually the UCI eSports Center, so it doesn't even look as cool as this. By the way, anybody in here happen to recognize this guy? Anybody know Michael Stevens, Vsauce? Any of the students? Oh, okay, one there. Yeah, two. All right, so in any case, if you want, you can see a whole thing on this on YouTube Red. In any case, we're going to then go and try to see if brain games, well, brain games is the wrong word. What brain games look like is they look like something like this. You'll subscribe to some package, or you'll do some free ones online, and they show you things like, here's a little checkerboard, here's a D, here's an M, and oh, here's a D again, so you should say, yes, I saw that one thing back. And you do this over and over and over again, that same task. Well, it turns out if you do that, you actually can get a lot better at doing that task, and sometimes it can actually transfer to some other kinds of tasks that use this thing we call working memory. But honestly, I can't last very long when I try to play these games. Even though I know maybe it's going to do something for me, maybe with lots and lots and lots of practice, it bores me to tears, I'm sorry. That's what we normally think of when we think of brain games. We don't normally think of this. These are commercial off-the-shelf video games designed with huge multi-million-dollar budgets by people who are better at computer graphics than cognitive experimental psychologists like me. And they're engaging and they're fun and they're these huge open worlds that you get to roam around and explore in. And as you're roaming around and exploring, everywhere you're moving, there's new stuff to see and learn, the relationships between them and new things to do, to figure out. It's a gigantic world you get to live in that is different than the one you're in now, and that might be a really neat, enriching kind of experience. All right, so a really, really bright postdoc in my lab said, hey, Craig, why don't we actually try that and see if that works? So here's how that works. Now, I got to admit, when he first came into the lab and he laid out this idea, he'd been working before he came to my lab, he'd been working in a lab doing work in neurogenesis in mice with this guy, Rusty Gage, down in San Diego. And he wanted to shift and work in humans, and I knew he didn't even know how to work in people, so I said, fine, Dane, we'll do this simple little study here. It's never going to work, but you'll at least learn how to do research in humans, and that'll be something. So we started off by just going and asking a whole bunch of undergraduates here about the games that they play, and then we gave them my very sensitive hippocampal-based memory task. And it turns out that those who played big, complex 3D spatial games did a whole heck of a lot better than those who didn't play games. Okay, Dane, maybe you do have something here, but there are a lot of confounds in this. Maybe these are the cool kids and these are the less cool. I don't know. There are a lot of things that could be confounds inside here. And so we're not going to know, as good scientists, we're not going to know if this is true until we try to actually move it ourselves. We have to do a real experiment. So here's that experiment worked. Take about 75 people, have them come on into the lab. These are non-gamers. And they got assigned randomly to one of three different conditions here. Either one case, no game control. We test you at the beginning, and then for 2 weeks, you just go and lead your life. I don't care. Do whatever you normally do. Other two groups played games. One of them, nice and highly addictive and entertaining, but not so much in the way of novelty. It's Angry Birds. Yes. Honestly, I have to go and give talks, big scientific conferences where I say, well, and in Angry Birds, you can tell that. But hear me out here. The other condition, Super Mario 3D World. Okay, look, this is a game, it's meant for kids, but there's a huge spatial component. You're roaming around in here. It's a 3D sort of fairly immersive kind of game. I'm taking non-gamers and I'm having them play this for 2 weeks. Here's what happens. I test you before, I test you after, playing a half hour a day for 2 weeks. If you are in the no game control, this is your before score, this is your after score, and this is your score after 2 weeks, aka absolutely nothing happened. If you play Angry Birds for 2 weeks, nothing happens. If you play Super Mario 3D World, wait a minute, you actually got better on my memory test that has absolutely nothing to do with plumbers or toads or anything like that. You get better on my hippocampal memory task. And it turns out that actually the amount that you explore the world and the better you do in it and the more of it you uncover, the more of an improvement in your memory. As if the more you were spatially exploring in that world and giving yourself something incidentally to learn because you're engaged, you're just automatically learning all of this stuff, that's improving your memory, kind of the way it does in the mice. Okay, you may be saying to yourself at this point, I mean seriously, are you sure this isn't a fluke? Dean LaFurla has a guy up here saying that Super Mario is going to actually make your memory better. Really? Okay, if you get past that, what is it about the game that could be doing it? Does it work in anything other than like college undergraduates here? And could it maybe help with aging, that whole thing before? Are there any changes in the brain? And I'm going to try to convince you that, well, yeah, no, it is real. So, okay, let's hit a little bit here. Let's try to first replicate it and see if we can do it in older individuals. So the way we did this study, this is a study funded by the Dana Foundation. I show up to your house after you've agreed to be in my study. I don't just randomly show up to people's houses. I show up to your house with a Wii U and a big flat screen TV, and you are assigned to one of three conditions: Super Mario 3D World, Angry Birds, or playing solitaire on your own computer. All right. So these, by the way, are 75-year-olds on average. Come on in. I give them the memory test at the beginning. All three groups are the same. If I have you play on your own computer solitaire for 2 weeks or a month, your memory's not gotten any better. Sorry, solitaire is not doing it for you. If you're the Super Mario 3D group in red, yeah, no, your memory's gotten better, just like we saw in the younger individuals here. Turns out also the Angry Birds group is showing a bit of an improvement, seemingly a little bit less than the Super Mario group. Now, the young folks, it didn't help. Okay, wait a minute here. Young folks have been playing things on there like phones. Even if you're not a gamer, you'll do a little bit, all that kind of thing. I'm showing up to a 75-year-old's house with a big flat screen TV and a Wii U, and it's like, oh my gosh, this is how I make it move, and oh wow, this is cool. And then that in and of itself is an enriching experience. And so it's quite possible that that's one of the things that's actually driving up even that group here in the older individuals. Oh, and by the way, sorry, let me not gloss over this. As we take a look at the size and shape of the hippocampus, it seems as if this Super Mario group at least showing good signs that their hippocampus has actually changed in volume a little bit as well. Not a massive amount, but a little bit there as well. But what is it about the game? Is it just Super Mario and we should all buy stock in Nintendo because that's going to be the prescription we're all going to have? No. It's this enrichment. It's leading this engaged lifestyle and giving yourself something new to learn and do. So here we took the game Minecraft, which, okay, is often played by little kids. One of the great things as a researcher that Minecraft does is it lets me completely control the world. So we come up with our own Minecraft worlds, and then we come up with our own things for people to do. And so in one case here, we give you a totally featureless world, but we give you a magical chest with any kind of thing you would ever want to build something with. And we just sort of treat it like a box of Legos. We say, okay, here you go. You got as many Lego kind of things as you want. Build stuff. Maybe start with the house, but then go do whatever you want. Now, some people may view this as the ultimate great thing in creativity. But have you ever watched kids with Legos? Yeah, you get the instructions, you build that cool little like TIE fighter or something like this, and then you take it apart, and when you're off on your own, you build a tower, you knock it down, you build the tower, you knock it down. There's a lot of that kind of stuff that goes on, and it turns out that they didn't really build a whole heck of a lot in the way of complex stuff if they were just left on their own to go do whatever they felt like. We anticipated that. Because we also had a condition here in which we said, okay, look, we're going to give you this flat, featureless world, and we're going to give you a magical box again, but we're also now going to tell you what's inside the box are exactly the parts that you need to build something cool. And as soon as you build that, we challenge you to build something cooler, bigger, tougher, more complex. To even build this, you have to do all sorts of stuff to get up that high, to place the blocks and all this kind of thing. So we're continually pushing you to do more and more and more and challenge yourself. We call this the directed building group. Another group here we just had, hey, look, here's Minecraft, go play. Try to get home each night. And that was it. And then the last group here, we sort of combined these two, go play and explore for a week, learn about your surroundings, come back each night, and then after a week we say, hey, by the way, go build this cool thing. And we're not going to give you the parts for it. You got to go out in the world and find them. So it was a really tough kind of task. They were really, really engaged in that. So here's what happens. On my memory task that, again, has absolutely nothing to do with Minecraft or any of this here, this group, absolutely nothing. Didn't matter. I kind of futzed around. I did a little bit. They weren't really all that engaged. They didn't do a lot. These two groups had a nice boost. That same exact boost, which, by the way, accounts for about 20 to 30 years worth of cognitive decline. That's the size of the boost that we're talking about here. So take 20 years off of your memory score. And then this final group here seems to almost be, it's not quite statistically, but it's almost here, twice as actually big as the other two. So the more you are pushing yourself to do new things, the more your hippocampus is actually getting better. And we can even see, remember I mentioned before there were those mice in the enriched environment, and I said that there were those who kind of moved around a bunch and explored the world a lot, and they got more neurons into their hippocampus than those mice who just kind of hung out in one little spot? Okay, well, we have some people that are like the mice who hang out in one little spot. This is an actual path that somebody took throughout the world. This person here stayed pretty still, had one big venture out, but besides that stayed in this area. This person over here explored a lot of the world. We can quantify that, and that's this measure over here. We call this exploration complexity. And it turns out that no matter which group you were in, the more you explored the world, the more improvement you have in this memory measure. So in all this kind of thing, really, are we sure? Yeah, we keep seeing this over and over and over again. 8 out of 8 conditions, 5 out of 5 experiments. Science has to go and be able to repeat, to replicate ourselves. We keep seeing it over and over again. What is it about the game? It seems to be pushing yourself to learn to do new things. And honestly, when you talk to people who study dementia, who study aging, what we've been saying for years is try to lead a cognitively engaged lifestyle. If you've been doing Sudoku for the last 20 years, it's probably not helping you anymore. Go learn something else. Do something else. And that's what this is telling us. That's the way to actually get at it. Does it work in anything other than college? Yeah, we've seen it. The average age in that study was 75. The range was 60 to 80. And we're having signs at least pointing to that this is actually making some changes in the brain that we can even see. All right, so take-home message 2 here from the talk here. Yeah, exploring these large immersive video game worlds can have very good positive benefits for you, acting as a proxy for this environmental enrichment, keeping you engaged and giving you something new to learn, breaking you out of your rut, getting you to do new things. Like rodents here, our memory performance improves with this enrichment and this spatial exploration. Okay, so I mentioned at the very beginning that none of this happens without students. That's how all of this actually works. I'm the one who gets to get up here and give the talks, but yeah, we have people that have been in the lab for a good long time. My wife and longtime colleague, Shauna Stark. We have people who have a Ph.D. already, like Dane Clemenson, who's in the lab, who's working on it. We have people who are getting their Ph.D. You saw some of the work that MR spectroscopy was Jessica Doce's work. And then we also have a whole host of undergraduates in the lab. This is about a quarter of the undergraduates who are in the lab I could manage to corral into the one shot that day. So this kind of thing, as I say, takes a team, and that team takes you all, from being participants in the experiment all the way down to actually helping us run it. And with that, I thank you very much and open it up to some questions.
Very nice talk, Craig. And we as all parents here have been trying to get our kids to stop playing video games, and now you just took away all the incentive that we have. So, any questions from the audience?
So hippocampus doesn't store memories, but it helps pull them from other parts of the brain?
So it's one of the most basic questions, and yet at the same time, we still don't have the perfect answer to it. It clearly is involved in really helping you store it. So when things are hitting you, one of the things we think that it does is it helps come up with a unique sort of code and a way to represent the information to keep it separate from other things. We call that pattern separation. One of the still fundamental questions is it just coming up with that code until letting other parts of the brain know how to treat it a little bit differently, and so the storage is happening there, or how much of that storage is happening directly inside the hippocampus? So we know that if you take it out, you'll forget everything that you learned for the last year or so. But you will still hold on to those things that you actually had learned several years back. Yeah.
A fascinating talk. I wished I could be in your lab in your class. So you list some of the campuses here. Let's say, for example, UC Riverside.
Mm-hmm.
I assume there are similar activities in other campuses of UC. So what are some of the leading programs, particularly from UCI, compared to other campuses?
Yeah. So for example, UC Riverside I'm listing right here. So this is Alana Bennett. She, a couple years before this, would have been like right back here in the, in the photograph, as she was a researcher in my lab who's now moved to set up her own lab at UC Riverside. And there, she's got an imaging center and she can collect data there, but she still works with us a lot. So one of the things, for example, that she works very heavily with, she works with Dr. Claudia Kawas in this 90+ study.